Hey, good morning. So today we are going to review for our first exam, which is this week. Um, and so hopefully that doesn't come as a surprise to you. As usual, when we have a chance to review like this, there's no way that I can, go over every single lecture, every single slide in detail. That's not really the point of this review. This is more an opportunity for me to go through some of the key slides taken from certain lectures uh, as a way to kind of remind you or to set up kind of like an outline almost of all the different topics that you need to remember to review on your own. So use this as kind of like a way of helping to organize your understanding of all the different topics that are important. And that way, when you go back and review your lecture notes, the actual lecture slides, maybe watch the YouTubes of the lectures again, you have a better sense of how to pace your studying. Okay. So this is a reminder then that we are responsible for lectures 1 through 11. So that's through last Friday's lecture. Today is the review session. So importantly, the first exam, it's going to be in 2 parts. Wednesday at normal lecture time. So this time on Wednesday, I need to fix the time. All right, sorry, I goofed right there. It says 10:30 to 11:30, but I'm going to change that because our class is 9:30 to 10:30. Okay, so I'm going to fix this, so don't be thrown off by that. Okay, so it's from 9:30 to 10:30 on Wednesday. You don't have to log into Zoom. You just need to log into your La Lima recitation section at that time and take your exam. All right, so it's not going to be proctored. Your TI will not be there. You just need to get on La Lima and take the exam. The exam will be available for a full hour from 9:30 to 10:30, but the exam itself is 50 minutes. So that means you sort of have like a little window of opportunity of 10 minutes or so to properly log in, get comfortable, and then you decide when to click start and start your exam. And from that moment that you click start and you begin your exam, then La Lima will time you for 50 minutes. All right. So that way you're not super stressed out about having to start right at 9:30. Okay. The format is multiple choice, the best choice A through D for each question. I think all of you have taken multiple choice types of assessments over La Lima already, so that should be pretty familiar. The one catch is, because this is being taken online and there's like 120 of you in this class taking it from home, from campus, from wherever. Obviously, there's no way that we can kind of control the environment to make sure that everybody is really doing it fairly. Because remember, this is supposed to be closed book, so no notes, no book, no internet, right? So in order to make this fair for everyone, the way that the multiple choice is set up is that once you answer a multiple choice question and you move on to the next one, you will not be allowed to go back. All right. So I know many of you hate that because you say in a normal semester, when you have a paper packet in front of you, right, you can skip over the ones that look hard or you're stuck on. And if you have time, you go back, right? But hopefully you all understand that in an online setting like this, we can't really allow that, right? Because then otherwise students, unfortunately, it's way too easy, right, for students to then, if they have extra time, go back and look up stuff on the internet, change their answers, or worse, get on the phone and take this as a group test, right? Hey, what did you put for number 10? That's not what I put. Okay, I'm going to change. No, right? So even though I agree it's kind of stressful, you can only answer the questions moving forward, and you cannot back up. All right. In the past, with 50 questions, 50 minutes, if you've studied hard and you know your stuff, you honestly do not need more than a minute to answer a multiple choice A, B, C, D question, right? If you know it, right? And this also cuts down on the whole double guessing thing, right? Because I think all of you have been in that situation before where you finish a little early and then you go back and you start double guessing, and a lot of times you erase the correct response and put the wrong one. All right, so keep that in mind. So that's part one. So that's this Wednesday. Everybody in the class logs on at 9:30, not 10:30. I'll fix this slide. And you take your test on La Lima. Then later this week, either Thursday or Friday, when you go to your normal recitation section, you will log into Zoom and join your recitation section as normal so that your TI can be with you. He or she can see all of you in the section. You need to have your web camera active, right? Because that's how this is going to be proctored, so that your TI can see you. If you're worried about your built-in webcam on your laptop, you can do this thing where you have your cell phone and log in to Zoom that way, so that you can set your cell phone next to you on the desk so your TI can see you that way while you take the exam in La Lima, right? So once your TI sees all of you and you're all ready, he or she will start the free response part during recitation. And that's also over La Lima, but it's just like the homework assignments. They're short answers where you have to type in a response. Okay, so it's just like how you guys have gotten used to doing the weekly quizzes with your TI, right? And then you will have the full 50 minutes of recitation to complete that. Right? I will send out an announcement on La Lima talking about those details that I just went over with you, including, of course, emphasizing that there's that typo on this slide. It should not be 10:30 to 11:30 on Wednesday. It should be 9:30 to 10:30 on Wednesday. Right? And the last thing I wanted to remind you is this week you do have a homework assignment due. I felt that it was worth it to give you guys a homework assignment to be able to have you guys practice the last set of material that you are responsible for on the exam, right? But because you're worried about studying for the exam, your homework assignment will not be due at the beginning of recitation this week. Your homework assignment will just be due by Friday night, all right? So that you have some extra time to do it. Of course, you benefit the most if you do the homework before you take the exam, right? But don't forget to turn in the homework. All right, so let's start reviewing the material here. So this first third or so of the semester, we have been introducing our study of the four main macromolecule families that make up life on Earth. Right? So we have our polysaccharides, our proteins, our nucleic acids, and our lipids. Remember that 3 out of the 4 of these represent examples of long chains of repeated subunits. And these are our polymers. And so those subunits are things like monosaccharides that are our basic simple sugars, our amino acids, which are our starting building blocks of proteins, and our nucleotides, right, which are our individual units that make up our nucleic acids like DNA and RNA. And lipids are the fourth example of macromolecules, but we don't really consider these polymers because there's no real discrete little subunit that you're using to build these up from. They're just long chains of hydrocarbons, right? Long chains of carbons with hydrogens. But of course, that doesn't mean that they're any less important than the other macromolecules. So we started off looking at proteins, right? Proteins that are built up by linking amino acids. So remember that life on Earth utilizes 20 different amino acids chained together in different ways to make the proteins that we use, right? And again, quick reminder, it doesn't mean in the universe that there are only 20 amino acids, right? In fact, on Earth, there are many, many other examples of amino acids. It's just living things evolved to use these 20. So be familiar with the standard structure of an amino acid, the carbon that has an amine group and a carboxyl group, right? And then there's a hydrogen and there is an R group, right? And it's the R group that varies. Depending upon what R group you have, that defines which of the 20 amino acids you're talking about. Right? So remember, you do need to recognize these amino acid structures by sight. You don't have to draw them, but you need to be able to recognize what they are if you see one, and you have to identify, is it nonpolar, polar, charged, hydrophilic, or hydrophobic? Right? And why do we emphasize these things? Because remember, proteins, right, their structure is all based upon the chain of amino acids that are encoded for by the gene for that protein. And those choices of amino acids, once they're linked together and the whole protein folds up, these side chains and the properties of these side chains are going to have a tremendous influence on how that protein folds up and whether it works properly. So as a review of protein structure then, remember protein structure is on several different levels. The primary level of protein structure is just thinking about the actual amino acid identity of these different positions in the polypeptide chain. Right, so valine, alanine, isoleucine, for example. Remember that amino acids are linked together to create a peptide bond. Peptide bond is the example of how these monomers are made into a polymer in the case of proteins. Right? In the case of other polymers, the same principle applies. Something is going to link together individual subunits using some kind of a polymerization process that we call a condensation reaction. Condensation because a water molecule is released in this process, right? To create, in this case, a peptide bond because it's between the carboxyl group and the amine group of two neighboring amino acids. Right? If you were to shove a water molecule back in here and reverse that process, that would be hydrolysis. Right? You put water back in so that the two amino acids can come apart. So that's the primary sequence. Recall that if we were to disregard the R groups that are up here in the darker kind of blocks up here and just think about this polypeptide backbone, right? So that's what you see here. One chain of the backbone going down somewhere, and then maybe somewhere else, another part of the chain is coming up, right? You would just naturally have some interactions just by the polypeptide backbone reacting or interacting, right, with another part of the protein through these oxygen and hydrogen bonds. So you might get some secondary structures like these natural kind of zigzaggy, flat sort of regions, or more sort of helical regions, again, because in this case you have hydrogen bonds happening between just the polypeptide backbone, or these other sort of more ambiguous shapes, right? So these primary, secondary, and then finally tertiary structures of these things coming together are important to keep in mind in terms of the process of how the protein, right, is made and then how it's going to function. This is already a nice review also that remember this polypeptide chain is being put together by the ribosome, which is itself a big complex of proteins and ribosomal RNAs. Right? And remember that the ribosome is where these amino acids are brought together because they're brought there by tRNAs. So when we discussed three-dimensional tertiary structures of proteins, and remember there's another level beyond that, because you could have an individual protein that's working together with other individual proteins to actually form a complete machine, for example, like the ribosome, right? We would consider that, right, the quaternary structure of a protein complex. Right? So remember that mature proteins, many of them that function as machines to do different types of things, they have to have some sort of on and off switch to control them. A lot of times this takes place through post-translational modifications of the protein. So small little functional groups like acetyl groups, methyl groups, phosphate groups can be added to the amino acids, either on places like the amine part of the amino acid. So a tyrosine can have an acetyl group added to it and become N-acetyltyrosine when it's added to the amine group. Or these functional groups can be added to the R group of the amino acid. So in this case, a phosphate group added to the R group of serine, so it becomes phosphoserine. And we said that once you do that, that would potentially alter the tertiary structure of the protein, because now the original shape was just all due to the R groups, just being left alone by themselves, interacting. Now you start making some of these R groups a little different by adding different things to them, right? That changes the the different sorts of interactions, and that might cause the tertiary structure of the protein to shift. And in many cases, that shifting, for example, if this is the start of the protein and you add a bulky phosphate group to, let's say, a serine in here somewhere, that might cause the overall tertiary shape to shift around, revealing new three-dimensional surfaces that previously were not there. And these shapes, these unique shapes, might be the right shape to bind something else, right? Either a small molecule that this protein then is going to work on or handle, or maybe it's another protein that this protein will interact with. And this protein number 1 might then change the shape of protein number 2. Remember when we talked about things like the way DNA is organized in histones, post-translational modifications is a very good example of sort of controlling protein function there. We then looked at sugars, starting off with our discussion of monosaccharides. So remember, a sugar, it can be thought of as a molecule where for every carbon that you have, N, right, you also have the same number of water molecules, N, as part of it. So that's why a sugar is a hydrate of carbon, right, which is different than hydrocarbon, right? Hydrocarbon, which we will look at the next slide, simply means that the C's have H's attached to them, right? But if we're actually talking about water or the equivalent of water, H2O, then this is a hydrate of carbon. Right? And so a simple sugar, we can call a monosaccharide, an individual building block. They end with the ose suffix. An example is glucose, which would belong to sort of a general group of 6 carbon monosaccharides that we just call hexoses. Right? So here's the structure of glucose. Remember, glucose, there are 2 forms that are important in biology. One is the alpha glucose that has the hydroxyl group at carbon 1 pointing this direction. If we were to imagine this on the plane of a piece of paper or on the screen of your laptop, right, then the beta form has the hydroxyl group pointing the other direction, right? And remember, it's because even though we draw these as rings, the truth is these are 3-dimensional molecules, right? So there are different directions that these different groups attached to the carbons will tend to go in just to kind of get away from each other. So they're not just like crammed up against each other, both pointing in the same direction, right? But this has important biological significance because if the shape of this molecule is slightly different, right, that means whatever molecule is meant to interact with this has to be also the right shape to expect that the hydroxyl group is up or down. Right? So that's what leads to alpha glycosidic bonds and beta glycosidic bonds. Right? So in our bodies that make things like glycogen, so mammals, we link a whole bunch of sugars together, a whole bunch of glucose together in our liver as a way to kind of store extra sugar that's floating around in our blood. Like after we ate a really big meal, right? And we have extra sugar, our body's not going to just waste that, right? Because evolutionarily, you don't know when the next time is. You'll come across this nice piece of fruit or something that gave you all this sugar, right? So your body wants to keep it, but it doesn't need to have it all floating around in your blood. So it can store it by linking it together as glycogen, right? And these glycogen are made by these alpha glycosidic bonds. Likewise, starch in plants, like this Okinawan sweet potato, purple potato here, also starch, right? So our bodies have the right kinds of proteins, the right kinds of machines to be able to recognize this shape bond so that we are able to digest things like starch, or our body's able to break down the glycogen that we've created, right? And that would not be true if you were dealing with beta glycosidic bonds. For example, plants also link glucose together to make cellulose. But in this case, right, it's a beta bond. And that subtle little difference makes all the difference because the enzymes in our body, right, the proteins that work on breaking down polysaccharides, right, they're not the right shape to easily break down a beta bond. Right? And that's why things like the fiber in your diet, right, like the roughage that you eat in your vegetables, right, it kind of passes through you largely undigested. All right, so So we talked about proteins, we talked about polysaccharides, and we talked about lipids. So lipids are an example of long hydrocarbon chains. So here you have a hydrocarbon chain with a carboxyl group at the end, and together this would make what we call a fatty acid, right? A building block of a potentially more complicated type of fat molecule. So you just need to remember that hydrocarbons, these are more useful for long-term energy storage because for the same amount of space, right, by cramming together a whole bunch of carbons chained together like this, right, that's a lot more carbon-carbon bonds that represent energy, right, compared to if you had individual sugar molecules, right? Individual sugar molecules linked together would take up more space, right? If you have just the carbons linked together, it's all packed together and you get a lot more potential energy storage that way. But the downside is something like glycogen made up of individual glucoses, it's easy to break off the glucoses if you need them again, right? Whereas the hydrocarbons, you're going to have to really work on this more to really get the energy out of it. Remember also, besides energy, fatty acids are very useful and important for structural components of our cells, like our membranes are made up of phospholipids, right? Fats also serve the purpose as signaling molecules. So fats that are released in your blood can signal different cells to do things. And of course, fat is a good insulator. It helps us maintain our homeothermic status of our body. Right? So be familiar with the 6 classes of different types of lipids that we discussed. Some of them are obvious, like fatty acids, triglycerides, phospholipids. Some of them maybe aren't as obvious. Things like steroids or terpenes, right, are examples of lipids. So then we moved on to looking at our DNA, our nucleic acids made up of nucleotides. So recall that our nucleic acids are made of nucleotides. So this now represents an individual building block of a nucleic acid. And so it is made of 1 nitrogenous base, 1 sugar ring, and 1 or more phosphate groups. Right? So in the case of our DNA and RNA, you only have 1 phosphate group on it to make the nucleotide that's usable as part of building something like DNA or RNA. Right? But you can have diphosphates or triphosphates. Right? So technically those would still be considered nucleotides. They just wouldn't be in the form that you'd find linked together in your DNA. So you'll have to know that there are purines and pyrimidines to choose from. So the 5 nitrogenous bases, again, these are the 5 that are used by life on Earth, right? There are a bazillion possible infinite number of different small nitrogenous bases that would fit some sort of structural similarity to these, right? It just so happens that these are the 5 that life on Earth evolved to use. The sugars that are used are more specific, right? So you either have a ribose sugar or a deoxyribose sugar. The difference being the carbon at the 2 position, whether or not it still has an oxygen attached to it or not. Right? And then you have the phosphate group. So then this is the structure of our DNA. Remember that our DNA is a double alpha helix. Right? So there's 2 single strands that are complementary to each other. Complementary meaning the order of the nitrogenous bases in each of the nucleotides matches up across with its complement. So adenines match up with thymines, cytosines match up with guanines. And remember that one strand, the strands run anti-parallel in 5' to 3' direction. So the left strand here, the way it's drawn, the 5' end is at the top. We call it the 5' end because this is literally the end of the DNA molecule, right? There's no other nucleotide above this one. So the 5' end, the very last carbon that's kind of on the end there is the 5 carbon, the C5, right? So that's this here. Right. Then the 3' end, what's sticking out on the other end is the hydroxyl group off of the carbon number 3. Right? And so those are normally held together in the middle of the strand through phosphodiester bonds. And because of the angles of these bonds, because again, even though we always draw them on a piece of paper or on the screen in 2 dimensions, bonds, right, there's no reason why these lines would all be perfectly flat along the surface of your screen. In reality, there's different angles just because of the electron orbitals, right? So the natural angles created by the phosphodiester linkages between the nucleotides contributes to the spiral kind of structure of the double-stranded DNA, right? And that's vitally important because that enables the bases in the middle to be at the exact right position across from each other, so you can have the correct Watson and Crick hydrogen bonding between the A's and the T's and the C's and the G's, right? And importantly, the shape of the alpha helical spiral, right? It's not perfectly symmetrical, right? You actually have these sort of bigger major grooves along the surface and smaller minor grooves along the surface. Again, very important because proteins that are going to come and interact with the DNA have to be the right shape to fit the major groove or the minor groove, right? And potentially see the specific DNA sequence of bases in there that give the inside of the twisted ladder its own sort of unique surface, right, or shape due to the sequence of base pairs. So be familiar with this structure. Importantly, where things attach to each other. Again, you don't have to draw this from scratch, but if you were to see different parts of this and you were asked, where should this link up with this other part, you should be able to know that. Okay. So remember, our DNA in our cells, they are not just naked DNA floating around. They are organized through tight associations with certain proteins. And that's what we form, that's how we form chromatin, right? So chromatin is DNA that's packaged up and organized through associations with various proteins that do 2 important functions, right? One is it's able to physically get the tremendous huge size of our genome, our basically 46 separate, chromosomes, because 2 pairs, right, of 23 each, all squeezed into the teeny tiny nucleus of one of our cells, right? So that's one important reason it's all packaged up as chromatin. The other, of course, is that by having DNA wrapped around proteins, you have control over what parts of the DNA sequence are actually accessible at any given time to be read, right? So the more tightly DNA is wrapped around proteins, the less accessible the genetic information is. So for cells that don't need to be, let's say, transcribing certain genes at a given time, right? Because maybe those genes have nothing to do with what that cell's function is, right? Maybe those regions of chromosomes are all bound up very tightly as heterochromatin, right? Whereas other regions where there are certain genes that need to be expressed or transcribed, maybe in those places the DNA is sort of more loosely unwound from the chromatin. So remember, the fundamental building block of chromatin is the nucleosome, made up of these 8 histone proteins that make the histone octamer. So again, a wonderful example, right, of how you have proteins whose job here is to manage one of the other types of macromolecules, right? These histone proteins, their job is to actually work on the DNA and help organize the DNA and help manage, right, what parts of DNA are more loosely available, what parts of the DNA are more hidden, right? And again, I think this is an interesting thought. Where do these histone proteins come from? They came from what? The mRNA that's translated by the ribosome to make these polypeptide chains that fold up as these histone proteins. Where did that mRNA come from? It was the edited form of some primary transcript RNA that originally was read by RNA polymerase from the gene that codes for the histone protein, right? And that gene itself, right, the histone coding gene itself, would be wrapped around other histones that are controlling whether or not more histone proteins are allowed to be made, right? It's kind of this roundabout kind of crazy thing, but that's why it's all related. It's all linked intimately with each other, right? So here again is another example of histone post-translational modifications. So these histone proteins, their amino terminal tails stick out from the nucleosome. So here's a wonderful diagram here. The gray thing is the DNA, the 146 or so base pairs of DNA that wrap around this single nucleosome made up of 8 histone proteins, right? Pairs of the 4 histone proteins in here, different color, but each one of them, you can see there's examples of the tails of the histone sticking out. So when these get post-translationally modified, for example, acetyl groups or methyl groups added to certain key amino acid residues on them, in particular residues such as lysine, K, that causes the shape of the histone protein to change, to kind of get more comfortable. That's how I always think about it, right? And that would either relieve or release its grip on the DNA or potentially tighten its grip on the DNA. And so when you have a whole string of these nucleosomes, as the DNA is wrapping around them like a string of pearls, right? These nucleosomes then can be sort of moving apart from each other, revealing sort of parts of the DNA that are now accessible for other proteins to get to them, right? Certain nucleosomes might just fall off the DNA. So you might have longer stretches of DNA where suddenly you can have a lot of transcriptional activity going on because proteins can get in there and RNA Pol II can be making lots of copies or lots of transcribed RNAs from it, right? Remember that DNA, the way our cells use DNA, at any given time, right, our cell is either busy carrying out its job of being, let's say, a fully differentiated nerve cell or muscle cell or a gland cell or a hair cell, right? In which case, right, it's busy expressing all of the genes that make it a hair follicle cell or a muscle cell or a nerve cell, right? The majority of the cells that you see when you look at yourself in the mirror, right, are the product of these different types of cells making all these genes that they need to be what you see, skin cells and things like that, right? But if they're busy doing that, that process is not compatible with them at the same time trying to make perfect copies and duplicating the genome, right? So that's why we said at any given time, if you pick a cell in your body and you look at it, it's either going to be busy expressing whatever genes it needs to express to be that nerve cell or gland cell, or it's saying, okay, I'm not doing that right now. I need to focus on replicating my DNA because I've been told I need to go ahead and divide and become 2 identical cells. Right? So that's very important to keep in mind. So we first started off looking at the process of DNA replication. So recall that during DNA replication, the 2 strands of the DNA are separated, right? And one of them serves, or each one of these can now serve as a template to make another copy of it so that you can go from one double-stranded molecule, split it in half, and end up with 2 double-stranded molecules, right? Because that's our semi-conservative DNA replication. Each strand contains the same information as the other strand because of this wonderful idea of base pairing. So the strand that gets read in this picture here, we call it the DNA template strand. And recall that the template strand is going to be read or copied in a 3' to 5' direction. And it's copied by DNA Pol III. DNA polymerase, right, because it's a big enzyme complex, a big quaternary enzyme complex that makes a big polymer of DNA. So it's called a DNA polymerase. Right? And so the DNA Pol III can only read the template strand in a 3' to 5' direction. Right? And as a result, the growing DNA strand that the RNA Pol III is putting together itself is made in its 5' to 3' direction, anti-parallel. DNA Pol III can only function by adding the next nucleotide onto an existing nucleotide. That's why DNA Pol 3 cannot start from scratch, right? It actually has to start with something already there. Okay? So DNA Pol 3 will take these nucleotides that are floating around, around it here. In this case, these are not monophosphates, right? These are triphosphates that are floating around. So there's 3 phosphate groups on it, and these 3 phosphate groups, right, are used. They break apart. The DNA Pol 3 breaks the phosphate bond, and that's what provides the energy. The energy then, so that this phosphate group can then be linked on to the free 3' hydroxyl group of this very last nucleotide here. Okay, and then that's why the strand grows in this direction, right? And remember that it's all based upon whether by chance the next trinucleotide that floats in here is the correct match. So in this case, a thymine across the adenine, right? Because if this were something else, like a G floating in here or another adenine, right, this would not be stabilized by the proper hydrogen bonding long enough to be able to properly sit in that position for the DNA Pol 3 to do this reaction and add it on. Right? If it was the wrong nucleotide, it would just float away. Right? So it makes you think about, right, how, sort of like what a big messy soup is actually inside of our cells at any given time, all these molecules floating around, randomly bumping into each other. And when certain combinations come together, right, then you're in business. So review the DNA replication process that involves other types of proteins as well. Remember that our chromosomes, they don't start from the end to be replicated. They just sort of start from these places in the middle where the double-stranded DNA separates, right? Because along these different regions of the chromosome, you have these conserved sequences where it's very rich in A's and T's, right? And as a result, those regions of the DNA strand tend to be weaker and come apart easier than other regions that have more G's and C's in them, for example, right? Because of the 2 hydrogen base pairing between the A's and the T's compared to the 3 hydrogen base pairs between C's and G's. Right? So at these origins of replication, you have the development of a replicon, a bubble in the DNA. And right away, as the 2 strands are separating, those are the original dark strands, serve as the 2 templates here. You have these light blue strands, which are your new growing DNA strands. And remember, because DNA Pol 3 itself can only read the template in a 3' to 5' direction and can only make the new light blue strand in a 5' to 3' direction, right? On this strand up here, no problem. The RNA Pol 3 complex is cruising right along, right off to the left here, as this almost like a zipper is opening up. But on the other side, because the other template strand is in the opposite direction, 3' to 5' this way, DNA Pol 3 can only work on this strand from the left to right direction. So that means on this strand, you have to make the copy strand in fragments. Right? As more of it becomes available, and this is unzipping off to the left, more opportunities to make another fragment. And that starts by a small fragment of RNA, not DNA, but RNA that's laid down by a primase protein, right? Why is it that you can't be laying down little pieces of DNA with DNA Pol 3? Because remember, DNA Pol 3 doesn't work that way. It can only add something onto something that's already existing there. But RNA polymerases like this primase, they can start from scratch. In other words, they can go ahead, randomly bind here, and start plopping down RNA bases that are complementary to the DNA. So as a result, you get these Okazaki fragments. And then importantly, once these start joining up with each other, you got to get rid of the RNA. Why? Right? Because it's RNA, right? Because its sugar ring still has the oxygen at the C2. That means it's not going to encourage the same kind of normal double helical structure. So if you had little pieces of RNA in the middle of your DNA, you'd have your DNA in a nice little alpha helical structure until you got to this weird little glitchy part right? And then the DNA, the alpha helical structure would be kind of disrupted, right? We can't have that. So the RNA has to be removed, and then that little spot has to be patched up to make it continuous DNA. All right, the central dogma of cell biology. So that was all about those macromolecules. So then we're getting into now this idea of how it is that our cells actually make the products of these genes that are encoded in our DNA and how they get turned into things like the finished proteins. Right? So the central dogma or paradigm states that information flows from genetic material in the form of DNA, right, through an intermediate molecule, that's our RNA, and then eventually into the creation of the intended protein products. So remember, we have different forms of RNA. We have the messenger RNA, which represents the actual coding sequence that's determined by the gene that gets transcribed, right? And then that RNA gets edited down. So you have certain parts of the original gene transcript, the primary transcript, that doesn't actually show up or is not necessarily needed in the final protein, right? So you have intron regions that are removed, and then the exons come together. So this gives you flexibility. So from one original gene in the DNA, through this process of editing the primary transcript of RNA that comes off of it, you can make several different forms of messenger RNA, each that will eventually be translated into a slightly different variation of a protein, right? So you get more different types of proteins out of it. So that's the purpose of the messenger RNA. Right? And so the messenger RNA, the tRNA, the rRNA, right, they are all the product of being transcribed from the DNA using RNA polymerase, right? Then the messenger RNA will later be translated by the ribosome into the finished polypeptide chain. Right? So transcription in our cells happens in the nucleus, and then the finished edited mRNA is then sent out into the cytoplasm, where the ribosome then meets up with it. And that's where these other two types of RNA play a crucial role. The ribosomal RNAs, right, like the little golden ribbons here, right, they are tightly associated with a whole bunch of ribosomal proteins to make the ribosome factory. Right? And that ribosome factory is going to read the messenger RNA as part of translating it. Right? But it's these little tRNAs, right, that are actually going to do the job of reading the mRNA triplet by triplet by triplet. And in doing so, that determines the correct amino acid that then gets linked together to create the growing polypeptide chain that makes our proteins. So remember, transcription starts at very key positions along our chromosome. It's not haphazard, right? The RNA pol 2 has to be told exactly where to start and start reading the DNA to make a primary RNA transcript, right? So these regions are these regulatory sites, such as promoters, right? So these promoters are often right upstream of the transcription start site. So they coordinate the recruitment of this big RNA polymerase II complex to start transcription. So here, I like this little plush toy here. So here we'll pretend there's a promoter region in here, and that's what we see here in the DNA. So what's in here? It's simply different specific short DNA sequences, right? Different combinations of A's, T's, C's, and G's. Let's just say we'll color code them. So this green represents a particular sequence. This pink up here represents another sequence. And you have transcription factor proteins, right? Proteins that are just the right shape to fit in the groove of the DNA right there. And importantly, they will fit better if the specific DNA sequence in here, right, the little very subtle differences in shape due to that sequence of A's, T's, C's, and G that are there, right, are the right shape for the three-dimensional structure of the protein to really happily fit in there. Okay, these transcription factors build up on these different sites around the promoter, and they eventually form a pile that's the right shape to recruit the RNA Pol II complex. So then that fits here, right? Otherwise, the RNA Pol II would just kind of float around, bounce off the DNA, whatever. But now, it fits together here because of all these piles of transcription factors. And importantly, that shape of the pile of transcription factors puts the RNA Pol II in exactly the right spot that evolution intended, so that down here you can see the RNA Pol II can start transcribing, right, the template strand of the DNA, and you get the primary RNA transcript coming out of the RNA polymerase. So remember, our genetic code that gets read and interpreted eventually into an amino acid sequence is a triplet code. Right? Every 3 bases of the DNA, right, it's used in that way, right? So it's read 3 at a time. And so the DNA, the sequence here, gets transcribed into RNA, that then gets edited down to make the finished mRNA, and that then is going to get read by the ribosome through the process of translation to then result in the proper instructions of which amino acids should be put together to make the finished protein, right? So very important that you get this. This process of going from DNA to RNA is transcription, and that happens in the nucleus. Then the process of the finished edited mRNA being converted or translated into the finished polypeptide chain by the ribosome happens out in the cytoplasm. So just a reminder, you'll be given that table of the triplet code, right? So that you'll be expected, if you were given a sequence of DNA, not only would you be able to identify what the RNA sequence should be coming off of it, but also what the resulting sequence of amino acids would be in the final protein. Keeping in mind that the very first amino acid that actually starts off the polypeptide chain is methionine, right? Because sure, there's DNA sequence that goes off to the left here, right? But that's not stuff that ends up being interpreted into the finished protein. And likewise, you would eventually hit a stop codon here that would stop this chain of amino acids. But even though the DNA keeps going on, even though maybe even the mRNA keeps going on for quite a bit. So it's very important to realize, right, what gets translated into these amino acids. So we said that the tRNA is responsible for bringing the amino acids to the ribosome. So recall, the tRNA has 2 really important parts to it. It has the anticodon that has a set of 3 bases in it that determines what codon in the mRNA this tRNA will properly match up with. Right? So in this case, this is the codon, or the set of 4 codons found in the mRNA that's being fed through the ribosome that code for alanine. Right? And this is our degenerate triplet code. The first 2 positions in this case for alanine have to be G and C, right? But the 3rd position can be any one of the 4 RNA nucleotides. Right? And that can be read because this particular tRNA, right, the first 2 positions are a C and a G to match up perfectly with the G and the C over here. But the wobble position, because it's at an angle, can get away with reading the 4th position. Right? And remember, the other end of this particular tRNA gets an alanine linked to it. So that's the alanine that gets brought by this tRNA to the ribosome. So we review the anatomy of a ribosome. The ribosome has 2 huge parts to it, a 60S and a 40S, each one of which is a combination of protein subunits and rRNAs, ribosomal RNAs. Inside, you have the 3 positions where tRNAs go through. So review the function of the A site. This is where the tRNA that's bringing the next amino acid would come in. And if it properly has the anticodon to match up with the next codon in the mRNA, it'll be stabilized there, and it'll have the growing chain of polypeptides that are still held onto by the previous tRNA in the P position, right? That will pass that chain over to the tRNA that's in the A position. And so it's the chain itself that gets added onto the next little amino acid that got brought in to the A position. And then the unloaded tRNA can then leave the ribosome through the E site. Okay. So go through the process of translation. Right? Remember that we learned about initiation, elongation, and termination. And I want you, importantly, to think about, right, in a figure like this, where did this mRNA come from? What does it represent, right? And what has it gone through already to even reach this point of being an mRNA? And what are these different features on it in terms of relating to where translation starts and where translation ends? And then the very last thing we talked about is as proteins are being translated by ribosomes, right? Many times, so here we have a chain of ribosomes all reading or translating the same mRNA that had just come out of the nucleus, for example, and that's why you see a growing polypeptide chain coming off of it. I encourage you to go look at that Wikipedia animation that very nicely kind of puts these things together. You can find that in our lecture slides. Or at least the link to it. Remember, many proteins need to have their translation finished in the endoplasmic reticulum to encourage the polypeptide chain to fold up properly. Other proteins are okay just to be translated into the cytoplasm, right, where they will then be moved to different parts of the cell where they're needed. All right. Okay. So I'll go ahead, stop the recording.